Hello everyone. So today I am going to discuss the Biochemistry Paper 2 MCQ question which was conducted by the Rajiv Gandhi University of Health Sciences, Karnataka. Right. So let's start. So the first question is, which one of the following is a non-protein amino acids? So as you know, in the body there are two types of amino acids. One which is incorporated into proteins and other amino acids which don't have role in the protein formation like ornithine, citrulline, GABA, beta-alanine and homocysteine. So these are basically the non-protein amino acids. So here the answer would be ornithine, right? Next question, that is transaminations of oxaloacetate results in the formation of. So you can see in the transamination reactions, when there is transfer of amino group on the keto acids, it will form a amino acid. So here the keto acid is oxaloacetate. Now the on oxaloacetate, if the amino group is transferred, it will be get converted into the aspartate with the help of enzyme aspartate transaminase, and the cofactor is pyridoxal phosphate. So here the answer would be aspartate, that is aspartic acid, right? Next question. Persons with the sickle cell trait shows an increased resistance to, as you know, in the sickle cell anemia there is point mutation, due to which at the beta 6 position glutamic acid is replaced by valine, and there is shape of the RBC becomes sickle. So when there is a sickling of the RBC cells in the low oxygen condition, it disrupts the malaria parasites life cycle and the malarial parasites does not survive. So it offers resistance to the patient who have the sickle cell anemia. So here the answer would be malaria. Rather you can say the patient of the sickle cell anemia would have some advantage that they will not get malaria because malarial parasite do not survive in the sickle shaped RBCs. Which of the following compounds is synthesized from the tryptophan? As you know, tryptophan produces niacin, melatonin and the serotonin. So the answer would be serotonin. Next question. Salvage pathway of the purine synthesis, right? So in the salvage pathway of purine, adenine, guanine or hypoxanthine, these all are directly converted into the AMP, GMP and IMP by a single enzyme, such as from adenine to AMP by adenine phosphoribosyl transferase, from guanine to GMP by hypoxanthine guanine phosphoribosyl transferase, or in the short form we tell it HGPRTase. By the HGPRTase, guanine and hypoxanthine is converted into GMP and the IMP respectively. So here the enzyme would be HGPRTase. One more question is, deficiency of HGPRTase causes Lesch-Nyhan syndrome. Right. Next question. The function of restriction endonuclease is very simple question, that it cleaves the DNA at the specific sites. Right. Second messenger which mobilizes the intracellular calcium is through the pathway of IP3, and the DAC calcium level is regulated, that is inositol triphosphates and diacylglycerol. By this the calcium level is regulated, as it has been written in the box. A sugar phosphate molecule, that is IP3, binds to the calcium channels on the endoplasmic reticulum. Right, this increases the concentration of calcium on the cytoplasmic site, which triggers the cellular responses like smooth muscle contractions and the platelet aggregation. So here the answer would be inositol triphosphate. Right, now the biological target for the attack by the free radicals are all of the following except. So as you know that free radicals attacks on the polyunsaturated fatty acids, DNA and the protein, but it doesn't act on the glucose. So the free radicals doesn't have any role on the glucose. Okay, next question. Retinoblastoma. That basically it is a mug of questions. So retinoblastoma is basically a oncosuppressor gene or tumor suppressor gene which suppresses the tumor. Now the last question is, fasting blood sample is preferred for the analysis of only one case mainly, that is the lipid profile, because if you give sample after the food or after eating the food, then you will find that chylomicrons is present in the serum, right? As you can see in the picture, if you give sample after eating, then you will find this white serum, that is due to the presence of chylomicron. So the answer would be lipid profile. And if you assess the sample with this, then you will find an elevated lipid profile. That's why we uses the fasting sample for estimation of the lipid profile. Thank you.